Welcome to all. I am Ms.G.PRIYANKA, Assistant Professor of Biochemistry, A.D.M. College for Women, Autonomous, Nagapattinam. Today discuss about carbohydrates. Introductions of carbohydrates are major source of energy from our diet, composed of elements carbon, hydrogen, and oxygen. Also called saccharides, which made sugars are produced by photosynthesis in plants, such as glucose are synthesized in plant from carbon dioxide, water, and energy from the sun, are activated in living cells to produce carbon dioxide, water, and energy. Carbohydrate, glucose, simple carbohydrate, simple sugar, complex carbohydrate. Carbohydrates are sugars produce energy when consumed. Our bodies break down carbohydrate to extract energy. Carbon dioxide and water are released in the process. Glucose in the primary carbohydrate our bodies use to produce energy. Carbohydrates are classified as biomolecules. Simple carbohydrates are referred to as simple sugars are the accent sweet to the taste. More sugars than needed from the energy result conversions of these sugars to fat. Complex carbohydrates include the starch in the plant and food fibers known as cellulose. Introductions of carbohydrates. Carbohydrates are found in the surface of cells, which is act road signs older. Monosaccharide distinguished on the cells from another. ABO blood marked from the red blood cells are made up of carbohydrate. They allow to distinguish our body's blood type of the forensic blood types. Carbohydrates in our bodies provide blood clots. They are also found in the genetic material. Carbohydrates also can combine with lipids to the glycolipids or with protein rich glycoproteins. Carbohydrates are most affine to all the organic compounds to the nature. The plant is the energy from the sun used to convert carbon dioxide and water into the carbohydrate glucose. Many of the glucose molecules are made of two long chain polymerase starts to the store energy. About 60% of food our diet consists of carbon dioxide. Such ways of utilize the carbohydrate in food such as potatoes and rice. Other carbohydrate classified disaccharides including the starch, table sugar, and lactose milk. During the digestion and cellular metabolism, carbohydrates are converted to the glucose, which is activated in our cells, provided our bodies with energy and provide to the cells carbon atoms, building molecules of protein, lipids, and nucleic acid. In plant polymerase to the glucose, classified cellulose bonding and the structural formations of cellulose and other important uses. The wood of the for nutrient pages of notebooks and cottons in our clothing in made of cellulose. Classifications of carbohydrates. The classifications of four types of classifications: monosaccharides, disaccharides, oligosaccharides, and polysaccharides. Monosaccharides are a simplest carbohydrate. They cannot be breakdown to the simpler carbohydrate, and disaccharides consist two monosaccharides joined together. They can be split into monosaccharides. Sucrose, table sugars can be breakdown into the glucose and fructose. Oligosaccharides can, and then anywhere the 3 or 9 monosaccharides in it. ABO blood grouping are the oligosaccharides and polysaccharides. Polysaccharides are large molecules containing into 10 to more monosaccharides units. Carbohydrate units of concentrations of chain form to the branched carbohydrate flowchart. Then carbohydrate, disaccharide, and polysaccharide. The monosaccharide is including the glucose, galactose, sucrose, and reducing sugars of disaccharides, including the maltose, lactose, and sucrose. The polysaccharides are polymerase to glucose. The two types of glucose, the plant and animal glucose. They are plant glucose is amylase, amylopeptide, and cellulose, and animal glucose is glycogen. Carbohydrate with free carboxyl group in hemiacetate for have positive test to the benefic or pellic test. And then which two have been hydrolysis to the refer reducing sugars and non-reducing sugar. The two types reducing sugar and non-reducing sugar difference is carbohydrate with free acetyl or free ketyl groups, and non-reducing sugars is acetyl or ketone group, not free but include inside the utilized bond formations. They are a reducing sugars, hemiacetyl or hemiketyl forms, and non-reducing sugars is acetyl or ketyl group forms. The example of reducing sugars: glucose, fructose, lactose, maltose. Non-reducing sugars is example sucrose and glycogen. Then monosaccharides. One sugars unit, important simple sugars of glucose, fructose, galactose. Glucose gives energy to the body activity in primary of cells, is usually not found in diet produced for the starch. Monosaccharides, the two types monosaccharides, fructose and galactose. Fructose, fruit, honey, sweet taste of simple sugars. Then galactose to milk sugar. Some important of the monosaccharide. Glucose is the most apparent to monosaccharide found in nature. Glucose also known as dextrose, blood sugars. Then glucose is breakdown in the cells, produce the energy. Diabetics have difficulty getting glucose in other cells, which is we must monitor to blood glucose level regularly. Then galactose is found combined with glucose in the disaccharides. When presents milk product, monomers as monosaccharides is found to the some fruits and vegetables. Fructose is combined with glucose is gives sugar, sucrose and table sugar. Fructose to the sweetest monosaccharides are found in the fruits, vegetables and honey. Fructose can be breakdown in the energy into the body. The test of carbohydrate, the Benedict test and Pelling test. The importance of Benedict test. The Benedict test is usually determine the process of oxidation reaction that occurs with sugars. The Benedict test can be used to urine to determine the level of the glucose levels in urine. Then glucose in high level of glucose in blood, then which is the indicator of the diabetes. Characteristics of monosaccharides are simple sugars which recur to the digestion, quickly apparent to the intestines and blood and carried to the liver. The liver converted in the glycogen and stored. Disaccharide consists in two monosaccharides which join together. They can be split into the monosaccharides. There are three examples of disaccharides: maltose, lactose and sucrose. Disaccharides are a simple table sugars which linked with two simple sugars. Important are disaccharides: sucrose, lactose and maltose. Sucrose equal to glucose plus fructose. Example: sugar, brown sugar, sugar cane and sugar beet. Example of lactose. Lactose equal to glucose plus galactose. Example: milk sugar. Lactose is lost solutions of sweet then sucrose. Maltose is equal to glucose plus glucose. Starch equal to maltose glucose. Sugars alcoholic to such used to candies. Disaccharides come to two monosaccharides. There can be make disaccharides join to monosaccharides by biosynthesis. Glucose plus fructose equal to sucrose. Table sugar found naturally in plants, sugar cane and sugar beet, honeys. Then sucrose may be provided to plants. Sucrose, the brown, white or powder sugars. Glucose plus galactose equal to lactose, the primary sugars of milk in milk product. Many people have problems digestion in large amount of lactose. Glucose to glucose equal to maltose produced with starch breakdown. Use naturally informations, reactions and algal and beef manufacturing. Maltose. Maltose is known as malt sugar. It is found to break down in of the starch. Lactose. Lactose is known as milk sugar. It is found to the milk and milk product. Then sucrose. Sucrose is known as table sugar. It is most apparent to disaccharide found in the nature. Sucrose found sugar cane and sugar beets. Oligosaccharides. Oligosaccharides are a type of the carbohydrate formed when 3 or 10 simple sugars linked together. Sample small amount of the naturally made plants. Oligosaccharides can be many functions of including cells, then the cell bonding. The from examples to glycoprotein have to the important role of immune response, maltodextrins and cellular dextrins. The AB4 blood types, AB4 blood types for carbohydrate on red blood cells. They chemical marks of oligosaccharides, they contain either 3 or 4 sugar units. Polysaccharides. Polysaccharides are large molecule of monosaccharides that are connected to each other to the types of polysaccharides, storage polysaccharides and structural polysaccharides. Storage polysaccharides contain only alpha glucose units, 3 important on the starch, glycogen and amylopectin. Structural polysaccharides contain with only beta glucose units, 2 important ones, cellulose and chitin, then contains to modify the beta glucose units. Polysaccharides, starch, glycogen, fiber. Starch equal to glucose, the churned legumes and tuberous. Glycogen is animal food and storage in liver and muscles. Fiber, plant food, the vegetable, fruits, not digestion by human. Cellulose, maybe cellulose. Amylose and amylopectin, starch. The starch mixed on amylase and amylopectin found in the plant food. Amylose make up 20% plant stores long chain amylase. Amylopectin make to 80% of plant stores during the fruits. Fruits starts to hydrolysis to produce glucose and maltose which are sweets. Glycogen. Glycogen storage polysaccharide found to the animals. Glycogen is storage in the liver and muscles. When glucose needed, glycogen and hydrolysis in the liver to glucose. Glycogen. Glycogen is a polysaccharide that stores glucose in muscles. Is similar to the amylopectin, but more have branched. Cellulose. Cellulose contains glucose unit bonds. Then glycosidic bonds converted to the change 3 dimensional shape of cellulose. Then amylose. Source of carbohydrate. Source of carbohydrate, food source. The glucose, fructose, galactose, maltose, sucrose, lactose, starch, glycogen, cellulose. Then food source. The glucose, fruit, honey, fructose, fruit, honey, and galactose, milk, maltose, food source, starch, and sucrose is sugar cane, and lactose is milk product, and starch tubers and legumes, and glycogen is seafood, cellulose, vegetables. Functions of carbohydrate: energy supply of brain cells, and synthesis of substance, non-essential amino acids, glycoprotein, glycolipids. The metabolic of lipid metabolism, and provided the fiber in the diet. Functions of carbohydrate in cells: major source of energy from the cells, major structural compounds of plant cells. Immediately energy from cellulose, the storage energy to the glycogen. Functions of carbohydrate: energy supply of the body functions, essentials of oxidation and fat, protein varying actions, provide energy. Synthesis of non-essential amino acids are present in some tissues, and add followers to diet. Then nucleic acid, connective tissues mixed glycocytic of nervous tissues. Necessary of the functions is CNS. Digestion and carbohydrate. Digestion is three steps: mouth, digestion of mouth, digestion of stomach, and digestion of small intestine. Digestion in mouth. The two types of digestion: chemical and mechanical digestions and chemical digestions. The saliva, the chemical digestion: salivary gland, oral cavities to the saliva. Salivary contains enzymes, salivary amylase and salivary amylase hydrolysis starts to maltose and dextrin. pH level 6.8 to 7. Only about 5% of starch and breakdown in the mouth. Then oxidation of pancreatic amylase and industrial enzyme. Hydrolysis oligo and polysaccharides in the monosaccharide for follow. Starch pancreatic amylase to maltose plus isomaltose. Maltose and maltase reactions: glucose plus glucose, isomaltose to isomaltase, and glucose plus dextrin. Dextrinoid glucose, sucrose, glucose plus fructose, lactase plus glucose plus galactose. Dietary fiber. Fiber is a simple type of carbohydrate from natural to the plant-based food that not digestible in human. Thank you.